Good afternoon to one and all present here, and welcome to the fourth day of our online life sciences workshop, The Key to Sustainability. Yesterday, we had Dr. S.P. Tiwari ji and Dr. Poonam Sethi ji as our resource person. Dr. S.P. Tiwari ji covered his presentation on the topic of PGPR, probiotic for plants, and Dr. Poonam Sethi gave her presentation about biomagnification, food chain, an interface towards sustainability. Today on day 4, we have Professor Rajesh Sharma ji, Professor Biotechnology Department, Veer Bahadur Singh, Purvanchal University as our first resource person. And his topic would be Introduction to Process Biotechnology. And our second resource person of the day is Dr. V. Srimati, Associate Professor, Guru Nanak College, Maths Department, and her topic is Application of Mathematics for Sustainability in Life Sciences using Fuzzy Models. So I welcome our resource person, Professor Rajesh Sharma ji. I welcome our second resource person, Dr. Srimati ji. Thank you for taking out time and joining our workshop, sir and ma'am. I also welcome our Dean Sciences, Dr. Noor Jahan ma'am and Dr. Ram Narayan sir for joining in again today with us. And I welcome all the participants who have been our continuous support. Thank you, all of you. So we'll start off the workshop. I would now request Miss Nahida, a research scholar, to introduce our resource person. Miss Nahida.
Thank you very much, ma'am. Good evening all. I'm Nahida Arif, researcher in Department of Biotechnology, VBSPU. I'm so excited to have this opportunity to introduce our first guest speaker, Dr. Rajesh Sharma. As ma'am said, he's professor in Department of Biotechnology in VBSP University, Jaunpur, and has a vast experience of 22 years in teaching. He's a strong proponent of the advancement of biotechnology. Sharma sir has did his BSc from Uday Pratap Autonomous College, Varanasi, affiliated to Purvanchal University. He obtained his MSc degree from School of Biotechnology, Institute of Science, Banaras Hindu University, Varanasi. After that, he joined School of Biochemical Engineering of IIT BHU as a CSIR JRF. Meanwhile, he got selected as an assistant professor in Department of Biotechnology, Purvanchal University, Jaunpur, in year 2000. He obtained his PhD degree from MGCGV Chitrakoot, Madhya Pradesh. Currently, he is pursuing supervising PhD students in the area of bioprocessing and bacterial endophyte. Sir is also a prestigious author of number of research papers in many reputed journals such as Asian Journal of Microbiology, Biotechnology and Environmental Sciences, Trends in Life Sciences, etc. Some of his publications are Characterization of Soil-borne Protease Producing Bacterium, Profiling of Plant Growth Promoting Bacteria associated with Jaunpuri Giant Radish, Rhizosphere, etc. Apart from this, he has published many review articles and also has contributed in authoring and editing of several book chapters. One of them is Recent Advances in Microbiology series. Sir has presented many research papers in several national and international conferences. He is often invited as an eminent speaker in many symposiums as well as universities. Sir has guided 15 MSc dissertation students and awarded 1 MPhil and 1 PhD as of now. He has completed several projects funded by UGC and some of them are still going on. Apart from all these, Sir is member of many of the committee of university, such as he is the assistant coordinator of International Quality Assurance Cell VBSPU, member of NAC steering committee, coordinator of Solid Waste Management and Disposable Committee, VBSPU, nodal officer, president of Student Alumni Association, and the list goes on. The title of the sir's presentation today will be is Process of Biotechnology. We are grateful for his presence among us today. Without further ado, please join me in welcoming Professor Sharma. Over to you, sir. Thank you.
Thank you, Ms. Naida. Thank you for the wonderful introduction. Sir, you can start your presentation.
Thank you, Naida, for your kind words. First of all, I will take an opportunity to express my sincere thanks and gratitude towards our Honorable Vice Chancellor, Ma'am, Professor Nirmala S. Maurya, who is constantly encouraged to organize such type of events. And I am thankful to Principal Guru Nanak College, Dr. M.G. Raghunathan ji, Manjeet Singh ji Nayar, who is the General Secretary and Correspondent of Guru Nanak College, and convenal duo, Dean Faculty of Science, Dr. Noor Jahan ma'am from Guru Nanak College, and Dr. Professor Ram Narayan sir from Veer Bahadur Singh Purvanchal University, Jaunpur, and workshop organizer and coordinator Dr. Dali ma'am, and workshop organizing secretary Dr. Manoj Pandeyji. I am very much thankful to all of you for providing an opportunity to discuss my views according to this particular topic, process biotechnology, which is the a key success to the industrial microbiology, whatever you are producing at the larger level, whether the product may be alcohol, maybe antibiotic, maybe different type of solvent. That is the basically ultimately you have to face various type problem during the production process, during the purification process, and ultimately to I will focus my topic on this particular topic, and basically it's a brief introduction about the topic. I'm not going to detail into the mathematical expression of the topic. Can I share my slide, ma'am?
Yes, sir. Yes, sir. Please do.
Today's my topic is introduction to process biotechnology. What is process biotechnology? Basically, it's a part of fermentation technology and part of the biochemical engineering. And this is the what every industrial microbiologist face some certain type of problems during the scale up of the process. So as far as fermentation process is considered, which is the part of the fermentation process biotechnology, as far as fermentation process is concerned, fermentation process may be defined as point of view of the biochemist, and may be defined as a point of view of the micro industrial microbiologist. So both are differing in their views. As a biochemist who define the fermentation fermentation is basically what is taking place, that is at molecular level, what reaction are going on in this particular process, how many electrons are produced, how many protons are produced, and how many ATP are being produced or consumed during the process, or how many NADH or FADH2 molecule is being produced during the process. So this is what a biochemist bothered about the these type of consideration, while they don't bother about the money which are involved in the during the process of the production. But as far as industrial microbiologist is concerned, which makes the process economically viable or cost effective, that is their main concern. He is not bothered about how many electrons are transformed, how many ATP are being produced during the process. So he is not bothering about these such type of events, but he is bothering about the money, how the process which is proposed by the biochemist or developed by scientists, how this process or technology can be utilized for the production of desired product at a cost effective in cost effective manners. Generally, if we talk about the fermentation process, generally we think the fermentation process is generally anaerobic process which is carried out by the microbial process. But this is the what narrow definition of the fermentation. But in broader definition of the fermentation process, it is the extending from microbial cell to another cell, say for example plant cell or animal cells, which is generally used or cultivated on a larger scale for the production of different product. So there is certain difference between anaerobic respiration and fermentation process. So let me refresh you what is anaerobic respiration, what is anaerobic respiration, and what is the fermentation. So anaerobic respiration is basically when the terminal of electron acceptor is oxygen, this process is said to be aerobic process. When terminal electron acceptor in the process, that is an organic component, say for example methane, maybe carbon dioxide, maybe nitrate, maybe nitrite, maybe ammonia. This type of process is known as a anaerobic respiration, while fermentation is defined in narrow term, it is anaerobic process, but in broader sense it is defined as fermentation process, where electron donor is organic molecule and electron acceptor is organic molecule. That is the fermentation process, and most of the fermentation process is generally aerobic in nature, using the plant cell animal cells. You might be knowing that animal and plants are highly aerobic in nature, and certain microbial population that is the aerobic in nature. Only certain microbial population which restrict the fermentation process for the production of desired product in absence of the oxygen, that is the anaerobic respiration or fermentation. As far as microbial product is concerned, so microbial product slide is visible, ma'am. Is slide is visible to all of you?
Yes, sir.
Yes, sir. Yes, sir.
Okay, okay, okay,
Slide
okay,
is visible, sir.
okay, okay, okay. Now, microbial products may be defined in different manner on the basis of different criteria. Microbial product may be extracellular product or maybe intracellular product. When the product produced, when substrate goes inside the cell of the microbial population or microbes, that is process organism, that is ultimately converted into product, and that is the leach into the medium, surrounding medium. And for the recovery of such type of product, you have to simply separate the cell and you have to recover the product from the fermentation broth or fermentation media. And intracellular product, for recovery of such type of a product which is being produced by the microbial population, that is reside inside the cell, and ultimately for recovering such type of product, you have to lyse the cell, you have to rupture the cell using different techniques. Say for example, ultrasonication is the one of the most popular technique that is being used for the rupturing of the cell, that is using the mechanical wave. And similarly, microbial product may be either growth associated or maybe non-growth associated. So this type of product may be growth associated. Say for example, if the microbial cell or microbial biomass itself a product, that is the growth associated. More is the growth, more is the product. Say for example, baker's yeast or single cell protein. Single cell protein is a very good example of the growth associated product. And as far as non growth associated product, generally secondary metabolites are used, which is generally produced during the early or mid phase of the stationary phase. And this phase is basically comprised of two phases, that is one is known as the idiophase and another is known as the trophophase, and most of the secondary metabolite is being produced during this trophophase. And one example, one another classification of microbial product is that is high volume low cost product and low volume high cost product. So this is one of the very good example of such type of product is the insulin. Insulin hormone for insulin is generally used to treat the diabetic patient. For obtaining a milligram quantity of insulin or gram quantity of the insulin in the earlier days, what will happen is we will sacrifice the pigs and we obtain the insulin from the sacrificed pigs. But as thanks to the advancement of the technology, especially recombinant DNA technology, which makes the process easier and economically viable, what will happen during the process? Say for example, you have to so earlier it was obtained from the sacrificing the hundred or thousands of the pigs to just get the milligram quantity or gram quantity of the insulin. But the same quantity of the insulin may be obtained from the culture of the 1 liter of 2 liter culture of the bacterial cell. Ultimately, what will happen? You have to isolate a particular gene. You have to identify that particular gene which is encoded for the insulin, and you have to cut gene and clone into the suitable
The
vector.
slides are not visible.
Okay, I am again sharing the slides. Now it is visible.
Yes, sir. Visible, sir.
Yes, sir.
हाँ दिख रहा है अब
हाँ हाँ ठीक है ठीक है. Now this is with the advancement of the recombinant DNA technology, so ultimately you have to identify this particular gene which is encoded for the insulin protein, and you have to cut that particular gene and clone into the suitable vector and allow to express, and the same amount of the insulin will be obtained from the 1 liter culture of the bacterial culture or 2 liter of the bacterial culture. So this is the what impact of the recombinant DNA technology in the on the process of the fermentation technology or biochemical engineering or process biotechnology. So earlier this insulin was the high volume low volume high cost, because if the demand is more and supply is less, then cost will be high. So this is the Malthus theorem. And now this insulin is, that is the product produced by the US based company in the 1976, and that is known as the humulin. That is the first recombinant product produced by the recombinant DNA technology. So the insulin is the first recombinant product that is marketed in terms of the humulin. Insulin is now labeled in the form of recombinant product, and that is the very cheap product. Now, earlier it was very costly, now it is very cheap product. So this is the impact of the technology on the desired product. Now range of the fermentation process, as far as range is concerned, there are 5 major groups of commercially important process which is generally used for the production of desired product. Say for example, those that produce the microbial cells or biomass, that is the itself biomass is itself a product. And example is Baker's yeast, example is the single cell protein. And those that produce the microbial enzymes, microbial enzymes means the commercially important enzymes, say for example amylase, lipase, and proteases, different type of proteases. And those that produce the microbial metabolite, that whatever microbial, what is the microbial metabolite? Microbial metabolite may be primary metabolite or secondary metabolite. Here we can find only the secondary metabolite, that is the our interest of the desired product. And those that produce the recombinant product with the help of recombinant DNA technology, as I already said that insulin was the earlier, it was the produced by the sacrificing the pig. Now the same amount of the insulin can be obtained by culturing the 1 liter or 2 liter of the bacterial culture. And those that produce modify a compound which is added to the fermentation medium, and generally it is termed as biotransformation. The biotransformation may be carried out, say for example steroid transformation is commonly carried out by the bacteria, generally bacteria, and the name of bacteria is generally used widely used for the biotransformation process is Arthobacter simplex. So this Arthobacter simplex is widely used for the transformation of biotransformation. So steroid transformation, you might be knowing that steroid is a precursor of steroid hormone is a ring that is known as, popularly known as cyclopentanoperhydrophenanthrin ring. So phenanthrin ring is the precursor of the all type of the steroid hormones. Now as far as containment level is concerned, so we should know before dealing with the process organism what is the nature of the organism. So as for European Federation of Biotechnology, process organism may be broadly divided into 2 categories, that is genetically engineered organism and non-genetically engineered organism. Non-genetically engineered organism is located into hazard group allocation, and it may be categorized into 1, 2, 3, 4 category, and it is the containment level. One is the known as GILSP, and rest 2, 3, 4 level is the containment level of 1, 2, 3, 4 level of the containment level. And similarly genetically engineered organism may be plant cell, may be animal cells, may be fungal cell, may be bacterial cell. It may be again broadly classified into 2 categories, that is harmless group first category and potentially harmful group second category. It is further subdivided into smaller, for the small scale culture and for the larger scale culture, that is the group A and group B. And similarly potentially harmful group second may be again categorized in the same manner, that is small scale and for the larger scale. And larger scale group B may be categorized as B1 level, and that is again known as GILSP. And larger scale level may be confined as B2, B3 and B4 level, depending on their virulence or their pathogenicity power. So this generally we in our laboratory use GILSP, that is organism that is known as the grass organism, genetically regarded as safe or generally regarded as safe. And this process is known as GILSP, that is good industrial large scale practices, that is used as a grass organism or genetically regarded safe organism. And before dealing with our process organism, what should we know about the process organism? First parameter, pathogenicity should be known. The organism which are dealing with the dealing with the for the production of desired product, that is the we should know the pathogenicity of the organism, how the organic is pathogenic or non-pathogenic. Generally we in our laboratory generally use the non-pathogenic form, because our laboratory design is in such that we cannot use the B3 level or B4 level of the organism, that is highly pathogenic forms of the organism. Say for example, Mycobacterium tuberculei or Mycobacterium leprae, or a very recent example, that is the leakage of the laboratory. People are saying that it is the leakage of the from the laboratory in Wuhan, that is the coronavirus. So such type of experiment will be carried out in very isolated place, for the very far away from the population, where most of the population is residing. So it is generally such type of laboratory is built in the isolated place. Suppose agar, by chance if the microbial population is escaped from the laboratory, it cannot going to harm the population, human population. Now, number of organisms required to initiate the infection. So you should know how many population, because there is a term quorum sensing is generally used. If the particular suppose you have introduced a single cell of the TB bacteria, that is Mycobacterium tuberculei, can you get the infection? No, because certain population is required to initiate the infection and onset of the disease condition. Now, rate of the infection should be known, and similarly, route of infection should be known. Most of the pathogens entered, say for example, coronavirus entered via mouth, or entered via nose, or entered via eye, and there is a receptor cell which can where coronavirus can get bound and transmit their genetic material, which ultimately hijack the host machinery for replication or transcription and translation. And amount of or volume of process organism should be known. How much you are handling the process organism in terms of volume, in terms of weight, in terms of life culture. And similarly, medical aid should be provided on exposure. So generally we cannot avoid aerosol while handling such type of organism. Suppose if you are dealing with the spore culture, it may create aerosol, but aerosol cannot avoid, because the size of aerosol is very less. It may be falls under the size of 1 micrometer to 10 micrometer. So you cannot avoid the aerosol by although you are wearing the mask, you are wearing the goggles for the safety measures, but you cannot avoid the. So such type of infection, say for example, generally we produce the citric acid in our laboratory by using the fungus Aspergillus niger. So generally spores are used for production of citric acid in our laboratory. So sometimes these spores may be entered in our body through nose or through mouth or through eyes, and it may create various health problems. So such situation should be, before dealing with your process organism, you should know what type of treatment should be provided, or immediately you should consult your doctor for the exposure of the such type of process organism. Now, little bit about how a bioprocess is established. So there is a sequential manner by which we can establish a bioprocess. Say for example, plant take if you visualize the slide by taking the example of biochemicals or animal tissue or plant tissue, so ultimately you have to identify the particular chromosome where this particular gene is located, and that cut that particular gene and clone into the suitable vector. Say for example, E. coli is representing showing here. Here is the E. coli having the plasmid. It is a basically extra chromosomal material which are present in the periplasmic space of the bacteria, that is E. coli, and cut the plasmid with the same restriction endonuclease, and ultimately we have to put this identified gene into the particular plasmid, and ultimately it is cloned into the suitable vector, and it allows to grow and divide. What will happen during the process of division? Multiple copies of the desired gene are produced, and ultimately it may be transferred into for the small scale production, and further going to the benchtop reactor or pilot scale, and finally the larger scale of the production, and after production of desired product, you have to recover the product and package the product and quality check and market into the for the marketing purposes. So I would like to mention a small note here where red arrow is showing isolation characterization of the microbial culture. So here, so you might be knowing that the microbial process during the cultivation process, a lot of heat is generated during the due to metabolic activity of the organism, or due to agitation, or due to aeration of the culture. So ultimately, suppose your desired product is produced at say for example 35 degrees centigrade or 37 degrees centigrade. What will happen during the process? The temperature goes up due to production of the due to metabolic activity of the organism, because it's a respiring culture, and due to process variables, say for example agitation and aeration. Generally we aerate the sample for the to meet out the demand of the aerobic culture for the production of the desired product. Sometimes oxygen is limiting. If the oxygen is not supplied in sufficient amount, what will happen? It drastically reduce the productivity of the process. So this suppose here you suppose you select an organism, isolate an organism from the soil sample or water sample or from the natural sources. Ultimately you have to incubate the organism at 40 degrees centigrade. What will happen during the this and use this particular organism for the production of your desired product, so that cooling cost can be minimized, because it can this particular organism which is isolated at 40 degrees centigrade, which can stand with the higher temperature, and it does not going to reduce the productivity of the process. So this is what. And different type of media composition is used for the production of different product. Say for example, animal cell culture media is entirely different from the plant tissue culture media, and plant tissue culture media is entirely different from the bacterial media, and bacterial media is entirely different from the uh, fungal media. So these in media designing and media devising a particular media for a particular process is a first area of the biotechnology for the production of your desired product. Now example of bioprocess, say for example cell culture and fermentation process, generally therapeutic antibiotic products, growth hormone factors, or plate derived growth related factors, insulin factors. So these are the basically factors generally being used in the animal cell culture media. And similarly blockbuster protein, that is the remicade, which is produced by a uh, 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 by the method of the recombinant DNA technology, which is active against the tumor, that is known as the tumor necrosis factor alpha, and it is also used for the rheumatoid arthritis and Crohn's diseases, and the product is licensed for FDA approval, FDA approved in 1998. So this was at that time it was termed as a blockbuster protein. Certain antibiotic that is considered as magic bullet. There is a compound that is Taxol, which is obtained from the bark of the inner bark of the taxa tree. That is the main source of the Taxol, basic Taxol, which is being used for the treatment of the cancer patient. That is the it forms the basis of the chemotherapy. So this certain type of a example. Say for example, in very recent year, very recently it was dostarlimab, that is the it's a type of antibiotic which is generally used for the treatment of the cancer, and news flashes in very recently that dostarlimab can 100% effective against any rectal cancer. So now more clinical trial have to be performed, and it has to be carried out in the different phases of the clinical trial, and I hope this dostarlimab can be used to treat any type of the cancer. Now it is that is this dostarlimab can used to treat the rectal cancer patient, and that is 100% recovery without any side effect. So sometime there may be time may become where a magic say for example magic compound may be obtained for the treatment of various types of diseases. So this is the schematic representation of the fermentation process, how fermentation process is set up and designed. So there is a stock culture, and ultimate stock culture may be in the form of liquid or maybe lyophilized culture. And ultimately, you have, for reviving of the culture, you have to put the culture of a small amount of the culture into the revival culture medium, and there is a shake flask fermentation, and then ultimately you have to optimize various parameters in laboratory level. Then if all the parameter is optimized, then go for the pilot level. Ultimately you have to again optimize the parameter for the pilot level, because the level is entirely different, and various factors playing the important role to optimize to reduce the productivity. So ultimately whatever parameter is optimized at laboratory level, you should again optimize the same parameter, optimize at pilot level and production level. So there is a raw material. So this process is basically consists of you can view in the next slide. So this process may be consist of upstream processing, downstream processing, and so in between these upstream processing and downstream processing, there is a actual fermentation process. So during the upstream processing, you have to prepare the media, you have to prepare the inocula, different inocula, you have to prepare, you have to sterilize the glassware, you have to sterilize the plasticware. So these all the machine process come under the section of upstream processing. And once you prepare the fermentation media, production media, then add the inoculum. Once you add the inoculum, fermentation process is started. And after certain period of time, say for example alcohol production, generally we produce the alcohol in laboratory with the help of grape juice or with the help of molasses. So it will take certain time for the production of a product at optimum level. So lastly, after completion of the fermentation process, you have to recover the product. So it is as important as upstream processing or fermentation processing. So these are downstream processing. Ultimately, you have to recover the product. This I have already told you. This product may be either extracellular product or maybe intracellular product. Ultimately, you have to recover the product, purify the product, pack the product, and market the product. So this is the diagram showing the cell line development for the production of animal based compound or animal based specialty protein, which is being used for the treatment of various types of diseases. Now some scale up issues during the process of the upscaling of the, there is two process, that is scale up process and scale down process. So we will talk about the scale up process, and there are various factors which can greatly affect the productivity of the process. I will only confine here with the oxygen, because oxygen play very important role. By controlling the precise concentration of oxygen, you may produce the precise concentration of your product. And similarly, different type of operating variables, reactor operating variables, heat transfer. Generally, heat transfer, I have already told you, the heating is a common phenomena during the processing of this type of material. So heating is due to metabolic activity, and heating is due to variables, different variables of the reactor, say for example aeration and agitation. And similarly, foaming is a common problem. Foaming is a common problem during the process. So in earlier stage of the process, foaming is due to media composition, because media contain high amount of the protein, and if you shake the protein, it will form the foam. So because aeration is there for the proper mixing or homogeneous to create the homogeneous condition of nutrient, homogeneous condition of the product, homogeneous condition of the suspension of the cell in the throughout the entire vessel or entire reactor. Then sterility is the slide नहीं दिख रहा है क्या?
नो सर, नहीं दिख रहा
No,
है।
sir.
ना दिखे तो आप बता दिया कि मैं फिर से शेयर कर दिया करूंगा. So these are the some sterilization process, that is various methods of sterilization is available. Say for example, wet heat sterilization, dry heat sterilization, filter sterilization. Suppose you are dealing with the animal cell culture or plant cell culture, ultimately you have to incorporate the different type of vitamins, different types of hormone, different type of growth factor. So these type of materials are heat labile or thermolabile material. You cannot autoclave it, you cannot wet sterilize it, so that you can adopt a technique, that is filter sterilization technique. By using the different membrane pore size, you can have the sterile hormone or sterile vitamins or sterile component, different component of the media. So I will focus mainly on the oxygen, because oxygen is the very critical during the aerobic process. Suppose it is estimated that 20, sometimes 50% alone cost of the product is due to oxygen. Suppose, so generally pure oxygen is not supplied, it is supplied in terms of sterile air, because oxygen, atmospheric oxygen contain 20% by weight of the oxygen. That is why people are preferring the sterile air rather than the pure oxygen, because if you are using the pure oxygen, it may going to enhance the cost of the product. So ultimately, this oxygen provision of oxygen to the aerobic culture will cost around 20% of the total cost of the product, or sometimes 50% of the cost of the total product. So this oxygen is very crucial. Suppose your culture is requiring 1 ppm of the oxygen in the culture, that is the optimum demand of the culture. So why we should supply the 10 ppm of the oxygen, or 10 ppm or the 5 ppm of the oxygen? Because it is going to affect the cost of the process. So we little bit, suppose there is a diagram showing the reactor, and generally oxygen is supplied from the base of the reactor, and this provision of oxygen with various types of devices, that is popularly known as sparger, and this sparger may be porous type of sparger, or maybe orifice type of sparger, or nozzle type of sparger, and all these type of sparger have certain limitation, and the main limitation is the pressure drop, because choking of the pores by growing cells, or clogging of the pores by the growing cell. So a little bit about the substrate, how much oxygen is required in particular process. So there is a equation which is representing the CH2O and CH2, that is CH2 representing the carbohydrate and CH2 representing the hydrocarbon. So suppose you are going to devise a particular medium, what type of substrate should be chosen by the what type of substrate should be choose to produce the product at optimum level, keeping in view the provision of oxygen. So if you take the carbohydrate, it will consume 2 molar of the oxygen, and because all the biochemical reaction takes place in the milli equivalent quantity. And suppose you are having the hydrocarbon as a substrate, it will require 3 times more oxygen. You can see the equation 6.67 CH2O plus 2.1 oxygen, that will give the bacterial cell composition on dry weight basis, that is carbon 3.92, hydrogen 6.5, and oxygen 1.94, and 2.75 mole of the carbon dioxide and 3.42 moles of the water molecule is generated. And similarly, as far as hydrocarbon, it will require, you can see here, 7.14 CH2 plus 6.135 O2. So you can compare the oxygen demand using the carbohydrate as a substrate and using hydrocarbon as a substrate. So you can see here in carbohydrate, it will require very less amount of the oxygen, but if you are using hydrocarbon as a substrate, it will require very high amount of the oxygen, because carbohydrate is present in their high oxidation state, and to oxidize this such type of hydrocarbon, you will require very small quantity of the oxygen. But in compared to carbohydrate, hydrocarbon is composed, it is present in the very highly reduced state, or highly reduced form of the compound. So it may require very high amount of the oxygen. So certain so this situation was observed by the great scientist Darlington 1964, and the same situation was given by an equation, that is A upon Y minus B equal to C, where A is the amount of oxygen for conversion of 1 gram of substrate to carbon dioxide, water, and if nitrogen is there, then ammonia is present. And similarly, B is the amount of oxygen required for the conversion of 1 gram of cell to carbon dioxide, water, and ammonia. And why is the
Rajesh
cell
sir,
yield? Ah,
Rajesh, your slide is not visible again.
okay, sir, I will again share. And why is the cell yield? That is gram of the cell produced per gram of the substrate, and C is the gram of oxygen consumed for the production of 1 gram of the cell. So this is the stoichiometric equation generally being used for the calculation of the desired product when you are using the substrate carbohydrate or using the substrate hydrocarbon or many other products. So there are various equations, are that is all type of this type of equation is that is put forth by the several workers time to time, that is the empirical type. So all the so this fermentation process or industrial processing is a process specific. Suppose you have to design a process for the production of citric acid at using the aspergillus niger and using the different using the particular media. So this process is specific. You cannot produce the alcohol by using this particular media, or you cannot produce the different type of antibiotic this particular media. So all the fermentation process which is being for the production of which is being used for the production of the desired product is a process specific, and it is almost empirical. And there is another equation put forth by the great scientist, that is Johnson 1964. In the same year, Johnson 1964, they divide the equation y equal to 32 C plus 8H minus 16O divided by Ym minus 1.58. Y0 is the gram of oxygen consumed per gram of cell produced, and Y is the cell yield, that is a gram of cell produced per gram of the substrate, and M is the molecular weight of the carbon source. What are the carbon source using this particular substrate, and CHO is the number of carbon, hydrogen and oxygen per molecule of the carbon source. So this is the equation generally used for the calculation of the product using the different substrate. Now effect of dissolved oxygen concentration on the QO2 of a organism, microorganism. QO2 is basically specific oxygen uptake rate, and it is generally millimole of oxygen consumed per gram of the cell per minute. So this is what the specific oxygen rate. So there is a graph which representing the dissolved oxygen concentration on x-axis, and specific oxygen uptake rate on y-axis. So it is similar to the effect of concentration, substrate concentration of enzyme on enzyme activity. So this, if you recall the Michaelis-Menten equation in during the derivation of the Michaelis-Menten equation, so you can have effect of temperature on enzyme activity, you have effect of pH on enzyme activity. In these two situations, that is effect of pH and temperature, you will get a bell curved shape, bell shaped curve. That is, if you increase the temperature due to increase in the kinetic energy, there is a chance of collision may increase, ultimately increase the rate of the reaction. But if you assess the effect of substrate concentration on enzyme activity, so if you increase the substrate concentration, enzyme activity increases, but after certain time there is no further increase, and there is no further increase in the enzyme activity. So similar is the case here. If you supply the oxygen, you will ultimately what will happen if you increase the oxygen provision, that will specifically increase the specific uptake of the oxygen. But after certain time, what will happen? It will, there is no impact, there is no specific uptake of the microbial culture by although you are supplying the continuously supplying the oxygen. So this is the, there is a process, there is a point where x-axis, that is a maximum oxygen uptake rate, is when you draw perpendicular line on this x-axis. So this value is known as the C critical value. So ultimately what will happen during the optimum process production of the process, ultimately you have to maintain this C critical value up to their optimum level. Otherwise what will happen, if it is less than C critical, the productivity of the process is going to decrease. Or if you so that you have to for maintaining this C critical level above C critical level, you have to continuously supply the oxygen in terms of sterile air. So you can see here there is a amino acid production by using the Brevibacterium flavum, and C critical for this particular experiment, which is carried out by the Hirosh and Shibai 1980, and investigation of a of amino acid biosynthesis by Brevibacterium flavum. So in this situation, there is oxygen is required oxygen is less than 1 ppm. So this is the quantity required during the process. So why we should provide the oxygen 5 ppm or 10 ppm? I have already told you that this oxygen concentration is greatly affect the cost of the process. So you can see here there is a production. So you can see here there is a glycolytic pathway fed in that is combined with the TCA cycle. So you can see different intermediate. So you may classify amino acid on the basis of different criteria. Say for example, essential amino acid, non-essential amino acid, and in the similar manner you can classify the amino acid on the basis of their precursor. Say for example, pyruvic acid is the precursor of valine and leucine. So this pyruvic acid is the end product of the glucose oxidation via glycolytic pathway, and ultimately pyruvate is converted into the acetyl coenzyme with the help of enzyme pyruvate dehydrogenase complex, and this ultimately combined with the oxaloacetate and will form the citric acid. So I will show here only the key component where the amino acid is derived, not the whole metabolic pathway. So this glucose is converted into erythroid 4-phosphate, and this phosphoenol pyruvate, which is an intermediate of glycolytic pathway, which is the erythroid 4-phosphate, and phosphoenol pyruvate, which is the precursor of the phenyl alanine amino acid. And similarly, oxaloacetate family of the amino acid and alpha ketoglutarate acid family of the amino acid. This alpha oxaloacetate family of the amino acid is the precursor of aspartate, and this aspartate is the precursor of the lysine and threonine is the precursor of the isoleucine. Similarly, alpha ketoglutarate is the precursor of glutamic acid or glutamate, and this glutamate is the precursor of three other amino acids, that is the glutamine, proline, and arginine. So you can have a look here. You can see, so certain amino acids is derived from the glycolytic pathway. Say for example, phenylalanine and valine and leucine, and certain acid derived from the TCA cycle intermediate, that is oxaloacetate or alpha ketoglutarate. So ultimately what will happen in glycolytic pathway, there is no requirement of the oxygen, but in case of TCA cycle there is a heavy amount of oxygen is required. So if you say you can see in the right hand of the graph that is represented by the degree of oxygen satisfaction, and that is the relative productivity of the amino acid. So you can see here the amino acid which are derived from the glycolytic intermediate will require very less amount of the oxygen, while the amino acid which is derived from the glycolytic TCA cycle or Krebs cycle pathway intermediate of the TCA cycle will require very high amount of the oxygen, because this TCA cycle is a aerobic process. So you can see here glutamate, arginine, proline require around 1 ppm of the oxygen, and less than 1 ppm oxygen will favor the formation of leucine, isoleucine, lysine, threonine, phenylalanine, and valine. So in this way you can see the importance of oxygen. By controlling the precise amount of the oxygen, you can control the precise amount of the different type of amino acids that is being produced by this Brevibacterium flavum. And for this, the critical value, that is I have already told you, it must be less than 1 ppm. So you have to ultimately maintain the continuous concentration, dissolved oxygen concentration in the fermentation broth is the 1 ppm, around 1 ppm. And similarly, another example you can see here, there is a cephalosporin C, which is considered to be the I think a fourth generation of the antibiotic, that is derived from the amino adipate, L-alpha amino adipate, that is LAA, will combine with the L-cysteine, which is the sulfur containing amino acid, and this will form the LAA L-cysteine, that is L-alpha amino adipate and L-cysteine. And addition of valine, this will form the LLD tripeptide. In the first step of the reaction, that is basically formation of the isopenicillin, and second step of the reaction, that is first step is catalyzed by isopenicillin N synthase, and second step of the reaction is catalyzed by expandase. It's a common name of the expandase, but enzyme is deacetoxycephalosporin C synthase, and third step of the enzyme is the deacetylcephalosporin C synthase, and it is commonly known as hydroxylase. You can see here, and here critical C critical value is the more than 20% oxygen. So if you supply the continuously supply, maintain the 20% saturation level of oxygen, you will get the cephalosporin C. Otherwise, if it is less than 20%, it will favor the formation of the penicillin N. So this is the how you can again see the example how oxygen is important for the formation of different type of product. If you maintain the oxygen more than 20% of the saturation level, that will favor the formation of cephalosporin C. If it is less than 20%, that will favor the formation of penicillin N. So this is the importance of oxygen, how controlling the precise concentration of the oxygen, you can control your desired product. Now little bit about the transport, or this is mass transfer of oxygen, concept of mass oxygen transfer. So this may be there is a common phenomena that is generally used for the at the larger scale level, that is continuously stirring of the fermentation broth. Why we continuously stir the medium? Because to maintain the homogeneous condition, and to maintain the condition of the oxygen, to maintain the homogeneous condition of the nutrient, and to maintain the homogeneous condition of the substrate that is being produced during the process. So ultimately what will happen, we have to design different type, and the main reason of agitation is to maintain the homogeneous condition. And the another reason of agitation of the medium is, suppose you are supplying the oxygen, it will generally in reactor it is supplied from the base of the oxygen, either via the porous disperser, or via the nozzle disperser, or via the orifice disperser. So ultimately what will happen, this oxygen goes up and rise. There is a bubble oxygen, a bubble of oxygen, a bubble of gases. What will happen, it goes up. It is because their density is very low, it goes up. What will happen, this larger size bubble is basically broken down into smaller size of the bubble, just to because larger size bubble is having less surface area available for the transfer of the oxygen. But you broken down this larger bubble into the smaller bubble, what will happen, this will ultimately provide the larger surface area for the transfer of the oxygen. And similarly, this is the different type of phases involved in the, so you can see this by visualize by this very picture. You can see here there is a gas bubble that is and there is a that is which are placed in the liquid media, and there is a microbial cell. This microbial cell may be either in single cell suspension, or maybe in mycelial culture, or maybe in pillated form. So there are the different types of resistances involved, which is just like electrical conductivity. More is the voltage, less is the resistance. High is the less is the voltage, more is the resistance. So that is the equation, that is V equal to 1 upon R. So you can recall the equation V equal to 1 upon R. Agar voltage, if you increase the voltage, resistance is going to be decreased, means electrons flow very freely. So the same situation is here. So during the transfer of oxygen, different type of resistances are involved, and ultimately this oxygen reaches to their target site, that is the microbial population, where actual oxygen can be utilized for oxidation of the product, for oxidation of the substrate, and ultimately converted into the product. And the first resistance you can see visually by this very picture, the bulk gas phase in the bubble. So basically there is a gas bubble, or gas bubble, which oxygen is diffused inside the bubble. Ultimately it will travel through the bubble, and ultimately reaches to the liquid media. So there is a interface between gas bubble, because gas bubble is surrounded by the liquid media. There is a liquid film throughout the gas bubble, around the gas bubble, and ultimately when oxygen is diffuses from the gas bubble to the liquid media, it will travel into the bulk. So there are various types of resistances are involved, and that is ultimately reaches to their target site, that is a single cell, or it may be pillated, or it may be flux, or flux of the cell. So what will happen? So there is a 7 type of generally resistances are observed during the oxygen transfer from bulk in a bulk media that is supplied from the base. And suppose there is a organism which is residing on top of the corner of the reactor. So ultimately this oxygen should reach at the top of the corner of the reactor. What will happen? Different type of resistances involved, and that is known as the mass transfer. So this is, there is a coefficient that is known as the mass transfer coefficient, and that is popularly termed as KL. KL is the mass transfer coefficient. So KL is nothing but it's a sum of the reciprocal of a different type of resistance. These 7 type of resistance involved during the mass transfer. So this can be explained by two film theory. This is the two film theory proposed by various workers to explain the how oxygen reaches to the their target site. So you can visualize here there is a two phase. One is the gas phase there, another is the liquid phase is there, and gas film, there is a gas film and there is a liquid film. So you can see here the C is the concentration of bulk in the gas phase, that's it, Cg, concentration of component C in the gas phase. Ultimately it reaches to their interfacial area where phase boundary is formed due to mixing of two boundary, that is the liquid boundary and the gas boundary. So there is a phase interfacial concentration which is represented by here in the gas phase, that is the CGI. CGI representing the concentration of component, gaseous component at the interfacial area, that is interface CGI, and similarly CLI, that is representing the interfacial area of the liquid phase, and ultimately it is transferred to the CL. So you can see here the concentration of CG, the oxygen concentration in gaseous phase is very, very high, but it is due to various type of resistances involved, the liquid the target site receive oxygen is the very, very less quantity or very, very low concentration of the oxygen quantity. So what will happen during the so I'm not going to detail of the mathematical derivation of this two film theory. That is, you can propose theory gas. So you can there is a different type of phases, that is the solid to liquid transfer, liquid to solid transfer, liquid to gas transfer, gas to liquid transfer. So if you combine gases, solid matter may be exist in 3 forms, generally 3 forms: solid, liquid, gas. So all type of combination is possible in industrial processing. So you may derive the equation for all type of the mass transfer from gas to liquid, or liquid to solid, or liquid to gas, because ultimately the receiver end is the solid particle, because microbial cell or animal cell or plant cell is the solid particle which is receive the gas which are present in the gaseous phase. So ultimately it will travel from the gas phase to liquid phase, bulk traveling to bulk liquid, and ultimately it is reaching to the solid particle, that is the process organism. So this can be explained by two film theory. Now, as far as solubility of oxygen is concerned, so if you compare the solubility of oxygen and solubility of glucose in a water solution, what will happen? There is a 600, oxygen is 6,000 times less soluble than the glucose molecule. So this solubility, as you might be knowing that if you increase the temperature, the solubility of solid will increase while solubility of gases will decrease. So that is why it will create the suppose there is a nearby industry which is releasing the hot water to the low lying area or river. What will happen? It will create a localized thermal pollution which is ultimately going to affect the biodiversity. Ultimately what will happen? The reduced in the biodiversity or lost in the biodiversity due to the localized thermal pollution. Now different factors which are affecting the oxygen demand, that is the rate of cell respiration. If the cell is highly respirable, then it will require more oxygen. And types of substrate, as I have already told you, if you use the glucose and methane, glucose will require less oxygen while methane will require very, very high amount of the oxygen, because glucose is present in their higher oxidation state and methane is present in their higher reduction state. That is why it will require very high amount of the oxygen. And type of the environment, suppose if you increase the temperature, gases will escape. In this situation, you have to supply the oxygen, you have to supply more and more oxygen, and there is a surface area or volume ratio that is known as volume to container ratio, that will explain in the next figure, in the next slide. So these are the some factors influencing the oxygen supply. What say for example, size of sparger gas, size of the sparger. Generally, it is estimated that if you are taking sparger size 1 mm, it will create the bubble size of 10 mm. So generally, the size of the sparger, pore of the sparger will dictate the size of the bubble. If you take the 1 mm pore size, that will create the 10 mm of the size, 10 mm of the bubble. If you take the 10 mm of the sparger size, that will create the 100 mm, a very larger bubble. And the design of impeller, size, number of blades, rotational speed. So these are the some factors which is greatly affect the distribution of oxygen throughout the media. And foaming is a common problem. Generally, it is avoided by adding the anti-foaming agent. Generally, silicon oil is widely used for control the foam formation. So sometimes foaming is not advisable or not added to the media, especially if you are dealing with the cell culture media, animal cell culture media. Anti-foaming agent is avoided, because it will going to uh, 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 create the problem during the recovery of the product. So in that situation, you have to install the mechanical foam breaker, and then that will time to time remove the foam from the system. And temperature, if you increase the temperature, it is certainly going to decrease the solubility of the oxygen. And type of liquid, so we will discuss about the liquid is basically two type, Newtonian type of liquid and non-Newtonian type of liquid, based on the Newton's viscous flow of the liquid. And gas hold up time, if the retention period, if the bubble reside for a longer period of time, it will efficiently transfer the oxygen. If there is if a bubble resided for a very less period of time, it will there is a very less efficient transfer of the oxygen. Now, determination of KLA value. How will you determine of the KLA value? So basically, KL is a one term and A is the another term, and the KL is the basically KL is the mass transfer coefficient, or A is the interfacial area available for the transfer of the oxygen, that is phase boundary between gaseous phase and phase boundary between gaseous phase and liquid phase. So basically, this KL value is a thumbs rule. There is a thumbs rule for the KL value. Higher is the KL value, higher is the efficiency of the reactor. Less is the KLA value, and there is a less efficiency of the reactor. Suppose if you are dealing with the very culture which is having very high respiration rate, or very high requiring oxygen rate, in that situation you have to choose the reactor according to their organism requirement. Now you can determine the KLA value. So there are various methods are available for the determination of the KLA value. That is a sulfide oxidation technique, and another is the gassing out technique. This gassing out technique may be further divided into two type of technique, that is static method of gassing out, another is the dynamic method of the gassing out technique. So here we take the example of sulfide oxidation technique. So this sulfide oxidation technique is carried out using a pure water, or pure water, that is distilled water. So if you take sulfide oxidation and you add once you added the oxygen into the medium, it immediately converted into the sodium sulfate. What will happen? So this oxidation will so you can get the slope, you can draw a diagram, and on x-axis that is time and volume of titrant, that is the sodium thiosulfate. So what will happen when you add at the time t0, that is when initial time, there is no formation of the Na2SO4, and once you supply the oxygen, it immediately converted into the sodium sulfate. And this unconsumed sodium sulfide is treated with the iodine solution, and it is back titrated with the sodium thiosulfate. So that is why it is the volume of titrant, that is the volume of the sodium thiosulfate, and it will against the time. So you will get a curve, and the slope of curve will give the value of the oxygen transfer rate. And what is the limitation of this? So there is a equation that is dCl upon dt equal to KLA C star minus Cl. So basically C star minus Cl is a driving force. If you recall this equation, what will happen? This, if you recall this equation, so you can see here the concentration of CGI and concentration of CI. When concentration of CGI equal to concentration of CLI, in this situation what will happen? There is no net transfer of the mass oxygen. So concentration will be just there is a statement कि stagnant water get rotten, जो ठहरा हुआ पानी वो सड़ जाता है. क्यों हो जाता है? यही इसका रीजन है कि there is no movement. Ultimately what will happen? There is no transfer of the oxygen, and water is having the very unique property, that is the self purification property due to the dissolved oxygen. So this will the sulfide oxidation, and this the limitation of this oxidation technique is it is very slow. It will take very long time, and it will depend on the fermentation rheology, because it is generally using the water. This particular KLA value will be determined by using the only water, not the fermentation media, which is actually different from the water. The there is a Raoult's law that is known. You know the Raoult's law. When you dissolve the impurity into the water, that will elevate the boiling point, and that is going to depression is the freezing point, and depression is the osmotic pressure. So what will happen during the formation of Faluda or Kulfi? People are adding the salt into the ice, because it will going to depress the freezing point. That is why Kulfi or Faluda is there. Cooling process is more effective in comparison to the ice alone. So driving force, so there is a gassing out technique, and this gassing out technique, what will happen? You can see here oxygen transfer. Basically the driving force in this situation is the C star minus Cl. C star is the saturation level, 100% saturation level, and Cl is the concentration of oxygen level in the fermentation broth. And so, if you plot a curve between the dissolved oxygen concentration against a time, you will get a curve like this, and the tangent of curve that is given by the y will give the value of the oxygen concentration at particular point. So this is not desirable, because it is one time determination of the oxygen. But in case of the industrial processing, ultimately you have to continuously monitor each and every parameter. Say for example, you are monitoring the pH parameter, you have to maintain the constant pH, you have to maintain the constant temperature, you have to maintain the constant dissolved oxygen concentration level. So this is a one point determination. So that is why it is highly discourageous in the fermentation process. And similarly, static method, so there are dynamic determination of gassing out technique. There are two methods, that is static method, and that is the another is the dynamic method. The only key difference between static method and dynamic method is the in static, ultimately you have to lower down the oxygen culture. So you can see here in the both the situation, that is static method and dynamic method. Ultimately in the static method you have to use liquid nitrogen to lowering down the oxygen. Ultimately if you supply the nitrogen, that will combine the oxygen, and this will give the lower down the oxygen concentration. And similarly in dynamic method, rather than supplying the liquid nitrogen, you have to use the microbial culture as a oxygen depleter. In this situation, there is a point X. What will happen? You have to put switch off the oxygen supply, and what will happen due to respiration or due to liquid nitrogen, oxygen concentration goes down. Once it is reached to the point B, you again resume the oxygen supply, and this will give the value of, so there is a slope AB which give the value of AB. The slope of this, you can see here, the slope is almost linear, while this B to C is the slope is somewhat linear, and ultimately it is the curve. So the slope is variable. Slope is not variable from A to B, because it is a straight line, but from B to C, slope is variable from point to point, and this slope will give the A to B give the value of respiration rate, and slope BC give the value of the oxygen transfer rate. So what will happen? So this dynamic method, in dynamic method, what will happen, there is a equation that is proposed by scientist Bartholomew. dCl upon dt equal to KLA C star minus Cl minus X CO2. So you can here there is a real picture of the fermentation condition where microbial microbial culture consume the process organism consume the oxygen, which is take specific oxygen uptake rate, and that is the millimole of oxygen consumed per gram of the biomass per hour. That is the QO2, that is a specific oxygen uptake rate or a specific respiration rate. So if you arrange the equation, that is for the Cl, you have to determine the Cl value, dissolved oxygen concentration. So you can just rearrange the dCl upon dt KLA C star minus Cl minus SQ2. So basically it is a non-linear form of the equation. Ultimately you have to transform the equation and we'll get the equation. Final equation is the Cl equal to minus 1 upon KLA into dCl upon dt plus XQO2 plus XQO2, and this will equal to Y equal to mx plus C. And if you plot a curve between dCl upon d2 plus xk2 and dissolved oxygen concentration in the fermentation media, and this will give the negative curve, and the slope of the curve will the value of the KLA, that is in the form of minus 1 upon KLA. So this is the some of the dynamic advantages and limitation of dynamic method. So advantages, it can be determined KLA during an actual fermentation process, and it's a very rapid technique and can use a dissolved oxygen probe of the membrane type. And limitation is the ultimately the it has certain limitation, and the main limitation is to it is applicable or it is implementable to a certain height of the reactor. Generally 100 centimeter of reactor, if you are using the height of the reactor is 100 centimeter, then this technique is applicable, valid. Otherwise it is not going to not going more than, if you are using the reactor height more than 100 centimeter height, this situation is not valid for the reaction. So you can see here aeration and mixing by this particular glass. So if you are using the, if you are generally you are carrying out the experiment in the laboratory with the same, so you can what is the KL value, you can by simply visualizing this figure you can transfer the oxygen transfer. The KLA decreases with liquid volume. So you can see in this first flask and second flask. First flask is having the more space because liquid is very less amount, and in the second step what flask there is a more volume of the liquid and less space is available for the oxygen. In this situation what will happen, KLA is going to decrease because insufficient provision of the oxygen, and similarly KLA is higher when baffle are present. So you will get if you are using the same volume of the liquid media with the same volume aeration space, what will happen, there is a plain flask and there is a notch flask which is having a buffer like structure, which is ultimately going to vortex the liquid, and that will increase the KLA value. And similarly there is a third situation where surface area is available. There is a first flask which is having the very less surface area, and there is another flask which is having the larger surface area. So larger the surface area, that is and there is a same amount of the oxygen, and that will give the larger value of the KLA. This is the their efficiency of the notched flask and with the in comparison to the plain flask, and surface area having the more flask having the more surface area will enhance the value of KLA. So this is the little bit about the, I am running late. Okay. So there is a fluid viscosity. The fluid viscosity will depend on the described by the rheological property of the, what is rheological property? The tendency of liquid to flow. If you take the water on a slide, or if you take a drop of water on a slide, and if you take a glycerol or honey on a slide and you tilt the slide, what will happen? Water flows very fastly, while honey and glycerol flow very slowly. So this is the rheological property, that is tendency of a liquid to flow, that is the rheological property of the liquid. And this is what described by this particular graph. If you plot between shear stress and shear rate, what will fluid viscosity? There is a shear stress or force per unit of area, that is the shear rate or velocity gradient of the plate rate. And this, if you plot a curve between the shear stress and shear rate, you will get a straight line, and the slope of the straight line will give the value of the viscosity or medium viscosity or density of the medium, where n equal to 1. There is a variation of this situation. So you can visualize here. So there is a variation from n equal to 1, that is for the Newtonian liquid, and if it is there is a pseudoplastic situation, there is a dilatant situation, there is a Cason body situation, and there is a Bingham plastic situation. So the example of Bingham plastic is generally we used different type of toothpaste, and clay is the example of the Bingham plastic of type of body. And Cason type of body generally is exhibited by the Streptomyces canamyceticus and Endomyces species. And similarly, dilatant rheology is not observed in the fermentation media, as it is represented by the cement and cement slurries representing the dilatant type of rheology. And pseudoplastic of rheology is basically mycelial fermentation. Example is the mycelial fermentation. So you can see here, so there is a deviation from straight line, and that is n equal to 1 for the Newtonian fluid. If it is less than 1 or it is more than 1, that is the deviation of the liquation Newtonian liquid, and it is the pseudoplastic, dilatant, Cason body, and Bingham plastic. So if you see the situation where what will happen during the, if you making the constant agitation with variable aeration rate, what will happen. So this is a figure A representing the non-aerated bioreactor. Once there is a constant agitation, once you increase the rate of, so this B, C, D, E, F is representing the increased rate of the aeration. What will happen if you suppose if you are having the constant rate of the agitation and aeration rate will increase, what will happen? There is a, you will get the situation F, where the all the oxygen which are supplied from the sparger, it is going outside the fermentation without any distribution. So this is what this situation F situation is known as the flooding phenomena situation, or flooding of the impeller. So this situation can be avoided by following this equation, that is Fs upon Ndq is less than 0.3 N square D upon G, where Fs is the volumetric air flow rate in meter per second, and N is the stirrer speed, that is per second, and D is the diameter in meter, and G is the acceleration due to gravity, that is meter per second square. So this is what flooding phenomena can be avoided by using this situation. So ultimately what will happen, you have to synchronize these values, that is aeration rate and agitation rate, in such a manner so that effective oxygen transfer can be takes place and your productivity will be optimum. So a little bit about this bubble driven reactor. So you know there are two type of, basically reactors are, bioreactors are classified in basically into two types of reactors, that is mechanically agitated reactor and non-mechanically agitated reactor. So these are the examples: bubble column reactor and airlift reactor is the mechanically non-agitated reactor. The circulation pattern will depend on the only their density, because gas there, if you supply the oxygen, so what will happen, it is mixes with the fermentation media. So what will happen, there is a gassed liquid and there is a degassed liquid. Gassed liquid is having the less density because due to presence of the air bubble, and degassed liquid having the higher density. So due to difference in their density, there is a circulatory pattern in such type of air reactor, that is the non-mechanically agitation reactor. So I have already told you there is a two parameter, that is KL value and another is the A value. So this is KL value is the volumetric mass transfer. It is a mass transfer coefficient, and basically KL represents the resistances, different type of resistances involved during the mass transfer of oxygen. So this KL is the sum of the reciprocal of the KL value, and A is the interfacial area available at the interface, that is the liquid and gas, where ultimately during the process of mixing, what will happen, ultimately you are going to decrease the value of KL and increase the value of A. In this situation what will happen, there is a increased transfer of the oxygen. So these certain factors which greatly affect the KLA value, that is agitation rate, aeration rate. As I have already told you, you have to synchronize these two values in such a way so that efficient mass transfer can uh, takes place. And cell morphology also plays important role. Suppose you are dealing with the suspensal culture, or you are dealing with the mycelial culture, or you will dealing with the pillated culture. So suspension type culture and pillated type culture both are generally behaving like a Newtonian liquid. And mycelial culture, because there is a mycelia, there is a branching in the mycelia. So ultimately, it is somewhat going to affect the density of the fermentation broth. So there is a rule. There is a mycelia. There is a branching also. So the degree of branching will depend the Ka will depend on the degree of the branching. More is the branching, less is the flexibility of the media. More is the branching, less is the flexibility, and more is the density, and vice versa. And cell concentration will also play very important role. जैसे if you take the example of cell concentration may be in the form of suspension and may be in the form of mycelial culture. And medium composition also play very important role, and substrate product concentration play a very important role to defining this KLa value. As I have already told you, there is two type of liquid, that is Newtonian liquid and non-Newtonian liquid. If you plot a graph between shear stress and shear rate, you will get the straight line, and the slope of this straight line curve will get the value of density of the media or viscosity of the media. So suppose there is a situation, you can imagine the situation, there is a suppose you are using starch or cellulose as a substrate. So
Sir, your slide is not visible.
just a minute. Now it is going to be concluded.
Okay, sir.
And Rajesh sir,
G,
you are also running sort of time.
just a minute पांडे जी, मैं conclude करता हूँ तुरंत.
Okay, sir.
Okay. So this is the suppose there is a situation, if you are having the substrate as a starch or cellulose, and you are producing different type of gums, say for example biopolymers, example say for example xanthan gum or gelan gum. So in this situation, what will happen? Because medium viscosity is very high, it is behaving like non-Newtonian liquid, and due to the progression of the fermentation process in the forward direction, what will happen? There is a hydrolysis of the substrate, and this viscous medium is converting to the less viscous medium. Now what will happen? There is a time it is converting to a Newtonian liquid, and at the time if you consider the product, because the product is itself a biopolymer, say for example xanthan gum or gelan gum, what will happen? It is again viscous medium. So it is starting from non-Newtonian tend to be Newtonian, and it is again converting to the non-Newtonian liquid. So this is what anti-foaming agent, you might be knowing this. Anti-foaming agent is generally surface active agent, which is ultimately modify the surface behavior of the fermentation media or fermentation bath, which is ultimately going to affect the KLa value, that is decreased KLa value if you are using the anti-foaming agent. That is why it is recommended that anti-foaming agent should not be sometime recommended. Say for example, especially if you are dealing the animal cell culture media or plant tissue culture media. So this is what all about a very brief introduction to the KLa value and process biotechnology, how a bioprocess is established, and how KLa value is important to the success of a particular process. So this is, I hope you will all enjoy this particular lecture. If any, every lecture has a certain shortcoming. If any shortcoming is point there, I apologize for that, and you have to point out me to further improvement our further presentation. Thank you very much. Thank you all of you. And again, I will express my sincere thanks to Dolly ma'am and Manoj Pandey ji, and our honorable Vice Chancellor and Principal Dr. M.G. Raghunathan ji, and General Secretary and Correspondent Dr. Manjeet Singh sir, and Convener Duo Nurjahan ma'am and Professor Aryan sir, and Dolly ma'am and Manoj Pandey ji, and many of our colleagues joined there, that is Dr. S.P. Tiwari ji, Manish Kumar Gupta ji, Rishi, and dear student from Visual Purvanchal University, Jaunpur, and from Guru Nanak College. Thank you all. Thank you all. Thank you.
Thank you so much, sir. Thank you. this is your area of expertise, sir, and it was wonderful hearing you. And I would like to let you know that we have students not only from Veer Bahadur and GNC, but all over India, students have joined us in this seminar.
It's
So
my pleasure to convey my views to all students who joined from all over India.
thank you, sir. So we would, of course, we will have another kind of workshop where we would like to hear you even more on some much better topics and your viewpoints, no extended viewpoints. So we will look forward for that also.
Surely, ma'am. Sure, sure,
Thank you,
ma'am.
sir.
It will be my honor. It will be my honor.
Thank you,
Okay.
sir. Thank you, sir. I will quickly call Miss Nahida again to introduce our second resource person of the day. Before that, I would like to welcome Vandana ma'am for joining us in the workshop. Thank you, Vandana ma'am, for joining us. Uh, Miss Nahida, can you
Uh,
introduce our second resource person of the day?
yeah, ma'am. Thank you very much, ma'am.
Yeah, thank
Uh,
you.
our other eminent speaker of the day is Dr. V. Srimati ma'am. She is the associate professor in the Department of Mathematics in Guru Nanak College, Chennai. Let me give you a very brief introduction about ma'am. Ma'am has completed her studies in mathematics and her area of specialization is fuzzy models. She has teaching experience of 13 years. Ma'am is known to have technical knowledge of SAP, MATLAB, and LaTeX. She even has advanced diploma in software technology and systems management from NIIT. Ma'am was a very bright student and thus is a gold medalist in MSc Mathematics. She was the head of Department of Mathematics from 2014, Dean Admin of Science Shift 2 from 2019, as well as Deputy Dean in 2002. She has organized several national seminars and workshops. Ma'am has conducted various competitions like maths quiz, art, models, puzzles, and paper presentation under banner of Gurumat in Shift 2. Dr. Srimati has attended various international and national symposiums, and she has also presented papers in them. Ma'am is author of many research papers in many renowned journals. Currently, she is attending several webinars, workshops, and FDP for mathematics tools as well as developing e-content. She has also been invited as a speaker for several webinars. Dr. Srimati's today's topic of presentation will be application of mathematics for sustainability in life science using fuzzy models. So without any further delay, let's welcome Dr. V. Srimati. Welcome, ma'am.
Thank you, Nahida. Thank you so much.
Pleasure,
Srimati
ma'am.
ma'am, we are waiting to listen to you. How do you relate Max Life Sciences? I was very, this thing that Max department is taking part in the life sciences. I really want to know now, ma'am. Welcome, ma'am.
Rajesh sir, kindly mute your mic.
Okay, okay. Thank you, everyone. Good evening to all. I thank the whole of Purvanchal University and Guru Nanak College, the Federal Secretary Correspondent, Principal Sir, Advisor Madam, Vice Principal Madam, and both the Dean Sciences of Purvanchal University and Guru Nanak College. And special thanks to Dr. Gauri, the organizer, for giving me the opportunity to be a research person this workshop. So today's my topic is application of mathematics for sustainability in life sciences using fuzzy models. The role of mathematics.
Ma'am, sorry, ma'am, I'm interrupting you. Can you just move the mic closer because the audibility is little low?
Now you are able to hear me.
Yes, ma'am. Better, clear, ma'am.
Okay. So the role of mathematics plays a very essential in daily life. Here I'm first starting and I'm going to explain about the Earth. The phenomenon on Earth is subject to mathematics, which is the only language we can describe them. So the mankind must favor mathematics into many approaches, which is more challenging, like climate change, protecting biodiversity, tackling the pollution, controlling epidemics, ocean sustainability, natural disaster, and man-made disasters. So Earth depends on the mathematical sciences. So it is not only helps us to understand the natural phenomena, also allows us to sustain the majority of the human activity on the planet. The transport networks, the internet, the business transactions, which are all practical applications of research, graph theory, and number theory. So finally, we could refer to it as a key role in education also. So mathematics is the basic education from the childhood. They learn mathematics. So together with their language or Tamil or English or whatever language they are into, the two pillars of which are the two pillars of the education system. So in our daily life, we use many devices which are all fuzzy logic, which we don't know what is the logic. So it is all the pattern recognition, air condition, then washing machines, vacuum cleaners, then braking systems, transmission systems, control of subway systems, unmanned helicopters, knowledge-based system for multi-objective optimization of power systems. So here I've just split all the fuzzy into few parts. That is introduction of the fuzzy sets. How we are using this. Already we know what is a set. We have learned in our childhood. Set is a collection, well-known, well-understood and distinguishable objects of our concept or our thinking about the collection. So here we define when we come to the fuzzy set. It is a collection of objects in connection with expression of uncertainty of the property, categorizing the objects by grades from 0 to 1. So X, suppose X is the whole universe, we said that universe itself is a set. Then we categorize from X, that is a function, from X to 0, 1. So we categorize everything within the 0 and 1. So we also have a function called the membership function, where there is X, mu of X, where X belongs to X, which is a fuzzy set. So in general, in real life, what is the fuzzy set? So we already come across many situations that inclusion and non-inclusion. We have lot of sets. We can split the whole universe into a set. So it can be included. Something will be included one set, some will not be included. That set will be included the other set. So for example, we have lot of class of tall people. We can split it into a set. Then beautiful paintings into another set. Then expensive cars. Then sunny days. So everything in the universe can be split into sets. So now we have come to this fuzzy set, like boundary of such sets are vague. So Lofty Zade in 1965, he proposed a complete new approach to vagueness called the fuzzy set theory. So an element belongs to a set to a degree k, which lies between 0 and 1. In contrast to the classical set theory, where an element must definitely belong to or does not belong to a set. So now we come to the definition of the fuzzy, proper mathematical definition here. If X is a collection of objects, then a fuzzy set A in X is a set of ordered pairs where A equal to X, mu A of X, where X belongs to X and mu A of X is a function from capital X to 0, 1. It is a closed set. So whereas mu A of X is called the membership function of A and is defined as a function from X to 0, 1. Here the membership function is given a grade of membership, that is, we know that the choice of different membership may change the set. So every set will have a membership function. So if it will take values only 0 and 1, and this is called a crisp set. So the analogies of the set theoretical notions of equality subset are defined as follows. The membership grade of each element of a fuzzy set in X is less than or equal to its membership. So first we go to the origin of the fuzzy logic. So it is from the ancient Greece, that is Lofty Zade. He found out the fuzzy logic, and it was then followed by the Professor Velachery-Tambaram in 1972, who organized the world's first working group on the fuzzy systems. And then in 1980, Smith and Company first was the person to market the fuzzy expert systems. So these are the history of the fuzzy logic, where the Lofty Zade, who was a faculty in electrical engineering, had a seminar paper in 1965 about the fuzzy set theory. Then first 1970, there was a first application of the fuzzy logic in control engineering. Then next it was in fuzzy logic was used in Japan, then it was in Europe, and then again in US. So logic becomes starting spreading out the worldwide. So fuzzy actually means it is not clear, distinct or precise or blurred. So actually fuzzy logic is a form of knowledge representation suitable for notions that cannot be defined precisely but depend on their context. So here where all we use the fuzzy logic, neural networks actually. How does a neural network work? It works like a human brain. So it is fuzzy.
Srimati ma'am, sorry to interrupt.
Yes, ma'am.
Ma'am, are you presenting only your first slide is visible, madam?
Oh,
Please, sorry.
not really.
Now I think, yeah. Yes, we can see the next
Now,
slide.
ma'am.
Yes, ma'am. We can see the next slide, and I think you can make it, no?
In the slideshow mode.
Yeah, slideshow. Are you able to see now, ma'am?
Ma'am, actually, it's in the slide mode only. You can do it at, from current slide. No, just play that. There's an option called from current slide on the top. Yes.
Now I can see.
It is still in the slide mode only, ma'am.
Not really. Now, ma'am.
The presentation. It is starting again, ma'am. 1 second.
Now.
It is in the slide mode. Click that from current slide, ma'am. That play button on the top.
No, ma'am.
No, it is not gone, but it's okay, ma'am. The visibility is there. You continue, please, ma'am. You continue. It is not going into the slideshow mode, but it's all right. The font is big, so it is visibility is there, ma'am. It is visible, ma'am. You continue, ma'am.
Ma'am, there is option slideshow in menu bar.
No, actually, ma'am is pressing that Shivam Kumarji, but it is not going into the slideshow mode. That is why she has been clicking on that slideshow button, but it is not going into the mode.
Yes, ma'am. Yes,
Srimati ma'am, it's okay. You start the presentation, ma'am. The letters were, the font was big enough, so we can, of course, it is clear to us, ma'am.
So now, fuzzy logic and neural networks.
Ma'am, your presentation is off. Your presentation is off, ma'am.
No.
Yes, ma'am. Presenting, ma'am.
You see me now?
Sorry?
You are able to see the.
Yeah, we are able to see the slide, ma'am.
Okay. So fuzzy logic and fuzzy logic in control system. Provide fuzzy logic in control system. Fuzzy logic provides a more efficient and resourceful way to control systems. So some examples are the temperature controller, anti-lock brake system, and here the temperature controller. We see the problem as we can change the speed of the heater fan based on the room temperature and humidity. So another way of controlling the temperature is temperature control system has 4 settings: cold, cool, warm, and hot. So humidity also can be defined as low, medium, and high. Using this, we can also define the fuzzy sets. So what will be the benefits of using a fuzzy logic? So we understand the physical system and control requirements. We can develop a linear model of um, plant sensors and actuators, determine the specified controller from control theory, develop an algorithm for the controller, stimulate, debug, and implement design. And we can also understand the physical system and control requirements, then design the control using the fuzzy rules, then stimulate, debug, and implement the design. So next we can also the fuzzy can be represented in the graph form. In the fuzzy terms, one representation for fuzzy number is about 600. And these are the definitions. So next we come to the example of a fuzzy model. So fuzzy models are being used in daily life without being aware of their presence. For example, we are using day-to-day washing machines, autofocus cameras, and so on. And fuzzy models are also models operate on information granules that fuzzy sets and relations are used. And this modeling is also realized at higher, more abstract level, and fuzzy models give rise to general architecture in which we highlight three main functional modules, that is input interface, processing module, and output interface. So the main mode of using the fuzzy models are input, granular input data, and fuzzy sets as output. So here also scenarios are here where we encounter collection of linguistic observations. So example, we have expert judgment, unreliable sensor readings, etc. So main categories of the models, we have an overview here. That is diversified landscape of fuzzy models. Selected category will be tabular fuzzy models, rule-based fuzzy models, fuzzy relational models including associative memories, fuzzy decision trees, fuzzy neural networks, fuzzy cognitive maps. Then first we have a mapping that we would have learned about mapping in our childhood. Like there is a two sets, one set is mapped to the other set. The same way we use here in the fuzzy relational map. That is FRM. It is a direct graph from or a map from a domain space to a range space with concepts like policies, events as nodes, and casualties as edges. It is also represents the relation between spaces D and R. So the directed graph from D to R denotes the casualty of D on R called relations. So every edge in the FRM is weighted with the number in the set 0 and 1. So when the nodes of the FRM are fuzzy sets, then they are called as fuzzy nodes. FRMs with edge weights 0 and 1 called simple FRMs. So FRM with cycles is said to be an FRM with feedback, where the causal relations flow through a cycle in a revolutionary manner. So the FRM is called here as the dynamical system. If the equilibrium state of the dynamical system is a unit state vector, then it is fixed point. So we have here to find out the hidden pattern of the fuzzy relational map. First we take a domain space and the range space. So D1, D2 is a domain and R is the R1, R2 will be the nodes. So here fuzzy we take two mapping, that is domain space and range space. Then E is a relational map. The hidden pattern was given when D1 was switched on and all the other vectors will be on. This is the concept for the fuzzy map. When one set is on, the other set of the range space will be mapped. So this was obtained by multiplying A1 into the relational matrix. So we are using here the matrix also, and A1 E1 is equal to R1 to Rm. After thresholding the vector A1 E1, vector B was obtained. Then B was passed into E1T to obtain B E1, and it is updated, and the threshold has to obtain the A2. So this procedure is repeated until the fixed pair was obtained, which gives the hidden pattern of the state vector A1. So this is the relational map which shows the attributes of the domain. Suppose we take a problem regarding this any person who is having a disease. So what are the attributes, like how to find out what disease they are having? Maybe they are having a headache and cold and fever, all that. We can take that all that as an attribute and relate it to maybe recently we have the COVID-19, or we can be a malaria, or it can be any other type of diseases which has the few attributes. We say that the symptoms as the attributes, and we relate to the disease. The disease we make it as the this will be the domain space, and range space will be the type of diseases. So D1, D2 will be the attributes, that is cold, cough, fever, or anything. And again we have a range, that is maybe it is whatever disease like thyroid or whatever. So we confirm, we take the symptoms and map it, and then find out what kind of disease it. Conclusion. So we map like this. D1, D2 will be the direct graph, and R will be the relation matrix. So this output of this will show the symptoms, and it will be only in zeros and ones. That's why we are using the fuzzy. Fuzzy only gives the output as zeros and ones, and finally we can find the solution for this. So hence the problem worked out in two distinct spaces, which gave a better conclusion that fuzzy cognitive mapping also can be used. Here we are using fuzzy relational mapping. Cognitive mapping also can be used, where the resultant vectors are on a binary state. So binary, we know the system can only understand zeros and ones, so it is on or off state. The node that results in on state, both in domain and range, that highlighted the cause and effect of the problem. So the attributes of the domain space were distinct to each other. Also attributes of the range space were disjoint to each other, and the cause and effect relations can be analyzed and using this fuzzy model. So these are the few fuzzy models, the fuzzy cognitive bimaps, where the directed map with policies, events is taken as nodes and casualties as edges, and the relationship and the concepts are used to find out the solution. Like FCMs with edge weights or casualties from the set -1, 0, 1, which is called as the simple FCMs. FCMs are fuzzy cognitive bimaps. This kind of model can also use to find out any solution for any problem. So another layered feed forward neural networks is also a computational structure that is inspired by observed process in natural networks of biological neurons in the brain. So we can take the attributes as one and domain and range, and find out the network into the neurons in the brain. Consists of simple computational units called neurons in the brain that are highly interconnected. So each interconnection has a strength that is expressed in the number, therefore to as weight. So the basic computational units of neural networks are artificial neurons. So we have input layer, hidden layer, and output layer. With this, we can input, and we can have the activation function threshold, and we can find out the output. Another one is the clustering, hard clustering, and which is where a set U is divided into clusters, and they are split into sets. The member function is to indicate the partition. So this partition, any problem can be partitioned, and solution can be found out. So the value of the membership function is taken as given as 1, and for a set X belonging to the cluster, the value of membership is given as 0. Then the important rule followed is should satisfy this relation. So fuzzy clustering, this downside of hard clustering has been rectified using the notion of fuzzy clustering. So if an element roughly between any clusters AI and AJ, it is impossible to calculate its membership value using hard clustering. So but in fuzzy clustering, membership value can range from 0 and 1. So the data set is fuzzy partition depending upon the degree of belongingness of the element in clusters. The membership graded value is given. So we have a few more fuzzy models, that is fuzzy transportation model, wireless network technology, decision making with fuzzy information, fuzzy optimization, and many problems are can be solved using the fuzzy. Since many problems are always vague, we have to find we have to find some solution for this. Also, fuzzy models can also be used for social and economic problems. Now we come back to the mathematics, where the how can we relate the mathematics for sustainability and how we can use through the fuzzy models also. So here, Max, how numbers, calculus, algebra, trigonometry can save the world. So there is a lot of research being done and understand how to improve the understanding of the climatic change. So the mathematicians are also um, doing some research on this. So they can also design better weather forecast and climatic models so that we can know what is happening beforehand. So accurate weather forecast can predict when and where extreme weather may strike, and climate projections are key to identifying weather patterns changing on longer time scale. So mathematician can play an important role, like working with a set of equations that describe the atmosphere, taking into account the temperature, pressure, humidity, and global circulation models also describe the interaction between oceans and atmosphere to look at what the average conditions could be in decades to come. And also mathematics, computer science, and the computers used to model the weather and climate, which is more powerful every year. Like Max makes these computers far more effective, both contributing to technology improvements in areas like quantum computing by algorithms where mathematics is used. For instance, and research is also being done for the weather, particularly how when storm occurs, and which is also the renewable sources like the carbon world by choosing the optimal location for wind farms, designing the effective layouts. Mathematicians ensure that these technologies harvest the maximum energy as effectively as possible. They can contribute to research in energy supply demand that ensures networks incorporate high proportion of weather dependent energy sources such as wind, solar, making sure that the lights stay on in years to come, and also preparing for the climate change. They also can use the understanding of probability and uncertainty to advise policymakers on likelihood of heat waves, floods, or other changes in weather patterns, help them to plan accordingly. So even in business, detailed information on how climate change might affect them. The food industry, for example, is highly dependent on agriculture and could use advanced warning of upcoming drought for instance to prepare themselves for smaller yields, and they also try to predict who might be at risk as they can prepare for the future. Also, collection of data can be done from ice sheets to cities, can deliver insights into the planet's physical process, human behavior, and everything in between. And the climate scientists rebuild the history of the planet's atmospheric composition by analyzing the tiny bubbles trapped in ice records in order to anticipate the scope of future changes. But without the statistical methods that mathematicians bring to analyze this data and assess its reliability, the information has less value. And the new technologies also can be developed, which is mathematical modeling is the key to the development of new technologies such as the CCS new technologies, be key to low carbon future, carbon capture and storage. So, for instance, to safely lock away the greenhouse gases emitted by fossil fuel power stations, and detailed mathematical models make this research possible by using the logistics methods, network analysis, statistical modeling, and many other mathematical tools. Also, mathematics can be made accessible to everyone. Max can't just save the planet on its own. Many of the global changes we face, like multidisciplinary, overcoming them requires mathematicians to collaborate with scientists and engineers in different fields. With a firm grasp of concepts such as uncertainty, probability, Mathematicians are uniquely placed to communicate the science data and forecast and ensure that this information is meaningful to the people who all need it. So mathematics have a real impact on our planet's fate. Mathematicians therefore need to communicate the importance of their work clearly and efficiently, effectively knowing when to swap the complicated equations for perceiving storytelling, pictures, games, or genuine interaction. Opening up maths up for the world to understand might just be the best way that we can come to our planet's destiny. So these are the fuzzy logics which have been used in the life sciences, where uh, we can find some solution for all this. It's just a type 2 fuzzy logic method for efficient data science models of effective medical informatics. Here, the general challenge in the medicine field is to provide the scalable image processing model which integrates the computing processing. And here, the fuzzy logic method is based on the fuzzy logic algorithm and and the high performance computing. So it is also assigned to implement the big data analysis and data science prediction models for healthcare. And also here the fuzzy stochastic Petri nets for modeling biological systems with uncertain kinetic parameters. Here it is also used to uh, have been widely used to model randomness in the feature of biological systems. For many biological system, kinematic parameters may be uncertain due to incomplete. So here we use the fuzzy models. Another adaptive neural fuzzy inference system for predicting protein activities. Then proposed model for disease diagnosis. Here we use so fuzzy logic for the immunodeficiency and tumor treated space. Then heart disease prediction using hybrid genetic fuzzy model. Then another one is the linear fuzzy network models obtained from microarray data by exhaustive search. Fuzzy modeling in orange production under different doses of sewage sludge and wastewater. Then profiling of medical equipment risk using fuzzy logic. These are my references. Yes. Able to hear, ma'am?
Yes, ma'am. Yes, ma'am. Srimati ma'am.
Yes, ma'am.
Yeah. So thank you so much, ma'am.
Thank you.
I could relate something. Of course, we would need a little more extended session to understand a little more on this. But yes, ma'am, thank you so much for bringing into light that every field, every aspect of science can relate to sustainability, including Max. So thank you for this presentation, ma'am. Thank you so much. Manoj sir? Manoj Pandey sir?
Yes, ma'am. Yes, ma'am. I am here.
Would you like to add something to this?
No, ma'am. Not ma'am.
Okay, Nuja ma'am is smiling. Ma'am, you have anything to say?
Yeah, madam has explained elaborately about the applications of fuzzy model. But as you said, we need
Yes,
some
ma'am. Yes,
extended time to understand this. Maybe a mathematic person can do justice like madam, but we need some time to, that is being a science student, that is a professor from chemistry. So I feel it a little bit difficult to correlate. Maybe we need to work about that.
Ma'am,
Thank
but
you, ma'am.
this idea of relating Max with life sciences, because I remember when we were talking and we decided to include Max also,
Yes, yes.
I was a little doubtful, but then Srimati ma'am was very confident that yes, life sciences
Yeah.
and Max has complete relationship. How will you do? I remember she telling me, how will you count? You count everything, right? So isn't that Max and life science? So yes, ma'am. Thank you so much for bringing this to light. That yes, life sciences is related to every aspect of our lives. be it any field, be it any subject. So thank you, ma'am. Thank you once again. Thank you, Noorjahan ma'am. Thank you, Ram Narayan sir. And I thank all the participants and all the professors who have joined us, uh, taking out time from their busy schedule. And tomorrow is our last day of the workshop, and we will be the people for the day would be Dr. Namashivayam sir, who is a former HOD of physics department, and he, when you listen to him, you will know the person with a very vast experience, and he's a wonderful dynamic speaker. And we have the other resource person. Sorry, Manoj sir, can you just help me in that, because I'm
Yes,
getting
sir.
into the
Professor Pradeep Kumar. He's the professor in Department of Biotechnology, Veer
yes.
Bahadur Singh, Purvanchal University.
And his topic is nanobiotechnology, the future of human health. Yes, so nanotechnology, the new happening of science which has been going on. So he'll be presenting his lecture on that. So we will see you tomorrow, but tomorrow the workshop would start at 3 o'clock, because since it's the final day of the workshop, so we will be having the resource people presenting their presentations, and followed by the deans summarizing the workshop for us. So it will be easier for the participants, if someone has missed a day or something like that, to understand that what actually the workshop went by. So we will start the workshop tomorrow by 3 o'clock. The flyer will be sent in, the timings will also be notified. So, but as a word, it is at 3 o'clock tomorrow. So kindly join us, and we will be posting the feedback link. Just give me a few minutes. So I thank all the participants for their time today, and I'm posting the feedback link. Kindly have a look.
And from tomorrow, we'll start to sending your certificates from
Yes, sir.
tomorrow onwards.
Yes, sir. Yes, sir.
Since registration is more than 400, then for per day 100 participants will get their certificate. It will take 2 to 3 days, 3 to 4 days to receive all the certificate from the for participants.
I have posted the feedback link in the chat box here. The YouTube also, the feedback link is getting posted. Participants, kindly check the chat box and pick up the feedback link, please, and start filling. The link will be open for half an hour. At 6 o'clock, we'll be closing the feedback link. So thank you.
Okay, thank you, Dolly ma'am. Thank you,
Thank you. Thank
Srimati
you so
ma'am.
much.
Thank you, all the profs. Thanks.
Thank you so much, ma'am. Thank you, ma'am. Thank you, ma'am. Manoj sir, we can wind up.
Yes, ma'am. We can wind up.
The feedback link has been posted in YouTube as well. So participants, please check the live chat box in YouTube and the chat box here in the Google Meet for the feedback link. Thank you.
Hello. Please share the feedback form.
Sorry, yes, ma'am.
Please send the feedback form.
It's in the chat box, ma'am.
Not shown.
You have just joined the meeting again, I think that's why.